हेरिडिटी और जेनेटिक्स के। तो ये पूरा व्लॉग उसी के बारे में रहेगा। सो आज हम इसके क्वेश्चन आंसर्स लर्न करेंगे। आपको दिखा देता हूँ एक बार। सो हमारा पहला टॉपिक सीधा हेरिडिटी पे है। व्हाट इज़ हेरिडिटी? सो whenever an infant is born in family, the relatives begin to wonder about the resemblance। ये फ़िलहाल इंट्रो है हेरिडिटी एंड वेरिएशन का। हेरिडिटी के बारे में resemblance on infant size, facial features, complexion, color of hair with those of the parents, siblings and grandparents. The source of such resemblance and differences are in the genes that are passed down from the parents to children and so on, generation after generation. This inheritance of genes is termed heredity. The study of heredity is genetics. The new individual develops according to the genes inherited from their parents. The transmission of characters from one generation to next, that is from parents to offspring, is known as heredity. तो इसके बाद इसमें हेरिडिटी का तो हो गया, अब वेरिएशन के बारे में पढ़ना है। अपार्ट फ्रॉम रिजेम्बलेंस, इट इज़ द फर्दर observed that siblings are same parents from same parents are unique and differ from each other except the identical twins. Such differences are termed variations. Variation means um, hmm, hmm. Variation means differences between parents and offsprings, or between offspring of same parents, or between members of the same population. So variation in a population is very important. It has a survival value for the population. This is because if the environment changes, some individual variant may be able to adapt new situations and save the population from dying out. So variation arises due to mutation or sudden change in the genes. Variation also arises because genes get shifted and exchanged during meiosis at formation of gametes, giving rise to new gene combinations. At fertilization, there is random mixing of parental chromosomes with different gene combinations. Such a source of variation, which is most common, is called recombination. Variation arises because of mutation and recombination. ये था हेरिडिटी और वेरिएशन के बारे में। तो अब मेंडेलियन इनहेरिटेंस के बारे में हम कुछ पढ़ेंगे। मेंडेलियन इनहेरिटेंस, सो जो सर ग्रिगोर जोहन मेंडल थे 1822 to 1824, he was an Austrian monk who used garden pea, Pisum sativum, for his experiments and published his results in 1865. His work, however, was rediscovered in 1900, long after Mendel's death, by Correns and De Vries. But since Mendel was the first to suggest principles underlying inheritance, he is regarded as founder of or father of genetics. We'll read something about Mendel's experiments. Mendel designed set experiments such that a pure tall variety of pea plants could be crossed to a pure dwarf variety. The anthers from flowers of tall plants were removed and their stigmas dusted with pollen from flowers of dwarf plants. The reverse experiment was also carried out. In the following spring, seeds from new plants were collected and sown, he found that all the plants of the generation called first female generation or F1 grew to be tall plants. He allowed them to self pollinate again. Then he collected seeds. The following year, after the seeds had been sown, he found that three quarters of these plants were tall and the rest dwarf. He repeated the experiment several times and found that the ratio of tall to dwarf plants was 3:1. In this way, he tried to cross pea plants with seven such contrasting colors. These were violet flower and white flower. Okay, pollen was exchanged between plants. Okay, tall pure breeding plants, dwarf pure breeding plants. All the seeds forward collected, sown होनी है. All the progeny grew tall. All the seeds were collected. Okay. So, what was the conclusion about these experiments? Plants with such. Now we have another question that mention the advantage of selecting pea plant for experiment by Mendel. So this is because Mendel selected pea plants to carry out his study on the inheritance. Okay, on the inheritance of characters from parents to offspring. He selected a pea plant because of the following features. Peas have many visible contrasting characters such as tall dwarf plants, round wrinkled seeds, green yellow pod, purple white flowers, etc. Peas have bisexual flowers and therefore undergo self-pollination easily. Thus, pea plants produce offsprings with same traits, generation after generation. In pea plants, cross-pollination can be easily achieved by emasculation, in which the stamen of the flower is removed without affecting the pistil. Pea plants have a short lifespan and produce many seeds in one generation. Okay. We have another question. Differentiate between the following: dominance and recessive, homozygous and heterozygous, monohybrid and dihybrid, dominance and recessive. So about dominance and recessive, we have some differences. A dominant factor or allele presents itself in the presence or absence of recessive trait. So the second difference, for example, tall plant, round seed, violet flower, etc. are dominant characters in pea plant. For example, in recessive dwarf plant, wrinkled seed, wildflower, etc. are recessive traits in pea plant. Now, there is a difference between homozygous and heterozygous. First of all, we have homozygous, which contains two similar alleles for a particular trait. That is, genotype for homozygous possesses either dominant or recessive, but never both the alleles. For example, capital R, or small r, it produces only one type of gamete. Now we have heterozygous on the other side. It contains two different alleles for a particular trait. That is, genotype for heterozygous possesses both dominant and recessive alleles. For example, capital R and small r, it produces two different kinds of gametes. Now we have monohybrid and dihybrid crosses. Monohybrid cross involves between parents which differs in only one pair of contrasting characters. For example, the cross between all the dwarf plant is monohybrid cross. Dihybrid involves cross between parents which differs in two pairs of contrasting characters. For example, the cross between pea plants having yellow round seed with those having green wrinkled seeds is a dihybrid cross. Then we have Mendel's principal law of inheritance. Basing on the results of his experiment, Mendel postulated the following laws of heredity. Law of segregation or purity of gametes. We have the first one. At formation of gametes, the two chromosomes of each pair separate, segregate into two different cells which form the gametes. This is a universal law and always during gamete formation in all sexually producing organisms. The two factors of pair pass into different gametes. Each gamete reveals one member of a pair of factors and the gametes are pure. Second, we have law of independent assortment. Law of independent assortment meaning whereby that in inheritance of two features, each feature controlled by pair of genes. Genes for two different features are passed down into the offspring independently. The segregation of one pair of factors is independent of the segregation of the other. So, the composition of the genes or genotype controls the outside expression which we can see, that is phenotype. The ratio of progeny in the crosses is therefore the phenotypic ratio. However, as more and more scientists began to devise genetic experiments, it became clear that Mendel's law does not hold true in all cases. So आज के लिए हम यही तक करेंगे, कल से हम इस क्वेश्चन से स्टार्ट करेंगे. So like, share, subscribe to the channel. Thanks a lot. And next video इस क्वेश्चन से स्टार्ट होगी. Thanks guys. अभी हमारा क्वेश्चन नंबर 6 रह रहा है. So Men explain the law of dominance using a monohybrid cross. Mendel's law of dominance states that uh, a dominant allele expresses itself in a monohybrid cross and suppresses the expression of recessive allele. However, this recessive allele for a character is not lost and remains hidden or masked in the progenies of F1 generation and reappears in the next generation. For example, when pea plants with round seeds are crossed with plants with wrinkled seeds, all seeds in F1 generation were found to be capital R and small r. When these round seeds were self-fertilized, both the round and wrinkled seeds appeared in the F2 generation in 3:1 ratio. Hence, in F1 generation, the dominant character round seeds appeared and recessive character wrinkled seeds got suppressed, which reappeared in F2 generation. Exception, codominance, incomplete dominance, etc. ये मैंने वीडियो में बोला था कि ये वाला क्वेश्चन कल होगा, लेकिन अब मैं और कुछ क्वेश्चन आज ही कर रहा हूँ. To define and design a test cross. Test cross is a cross between an organism with known genotypic and recessive parent. It is used to determine whether the individual is homozygous or heterozygous for a trait. If the progenies produced by a test cross show 50% dominant trait and 50% recessive trait, then the unknown individual is heterozygous for a trait. On the other hand, if the progeny produced shows dominant trait, then the unknown individual is homozygous for a trait. ठीक है. तो इसके बाद आता है हमारा using a Punnett square, work out the distribution of phenotypic features in between a homozygous female and a heterozygous male for a single locus. In Guinea pigs, heterozygous male with black coat color is crossed with the female having white coat color. The male will produce two types of gametes, B and small b, capital B and small b, while the female will produce only one kind of gamete, B, capital B. The genotypic and phenotypic ratio in the progenies of F1 generation will be same, that is 1:1. Okay. When a cross is made between tall plants and yellow seeds and tall plants with green seeds, what proportions of phenotypic in the offsprings could be expected to be tall and green or dwarf and green? A cross between tall and yellow seeds and tall plants with green seeds will produce three tall and green plants, one dwarf and green plants. Now we'll read about deviation from Mendelism. Since the rediscovery of Mendel's laws in 1900, cases have been reported which gave additional information regarding the patterns of inheritance. The following ideas of Mendel had to be modified. Mendel found that each of the seven characters considered was controlled by a single gene. Although gene is a functional unit at the biochemical level, character may be the result of coordination of functions of several genes. Therefore, for a particular character, there may be two or more than two genes responsible, and these genes may interact in different manners. This lethality and interaction of genes for each of the seven pairs of characters examined, it was observed that one allele allomorph dominated over the other, so that F1 exhibits uh, one or more other alternative phenotypes represented in the parents. However, soon after rediscovery of Mendel's laws, experiments were available to show that in some cases the F1 individual showed the phenotype which was intermediate between the two parents. For instance, in four o'clock plants, in each of seven pairs of characters studied by Mendel, there were only two alternative forms from each character. That means that two alleles were present for each character. This also led to a belief that for each character there were two alternative forms, one dominant over the other. Later work showed that for a character there can be several phenotypes. For example, for rabbits, the body color can be of four or more types. Therefore, concept of alternative allelomorphs had to be modified by the concept of multiple allelism. According to law of independent assortment, any two or more than two pair of characters assort independently of each other. Exception to this phenomena were discovered due to linkage and the associated phenomenon of crossing over. So क्वेश्चन नंबर 7 से हम कल स्टार्ट करेंगे. So